Online webinar on vaccine clinical trial, COVID-19 vaccine candidates. Hi everyone, I am Dr. Shivangi Singh, Program Director, Fineness Institute of Clinical Research, Delhi. Now firstly, before going into the vaccine clinical trial, we will discuss what is a vaccine and how do vaccine work. A vaccine is a type of medicine that trains the body's immune system so that it can fight a disease it has not come in contact with before. It is a biological substance designed to protect humans from infection caused by bacteria and viruses. Vaccines are also called immunization because they take advantage of our natural immune system's ability to prevent infectious illness. Vaccines are designed to prevent disease rather than treat a disease once you have caught it. So basically, you are given a small amount of harmless form of a disease, then your body makes antibodies to fight it off. Then if you encounter the disease again, your body already has the antibodies, so you don't get sick. You are now immune. Now we'll just briefly discuss antigen and antibody. What is an antigen? Any foreign signal that induces an immune response. Usually refers to the molecules of the cell surface that are recognized by immune system as foreign. Usually made up of carbohydrates and protein. Now antibody, also known as immunoglobulin, Ig, is a large Y-shaped, as you can see in the image, Y-shaped protein produced by plasma cells that are used by the immune system to identify and neutralize foreign objects like bacteria and viruses. Each antibody recognizes a specific antigen unique to its target. Now we'll just see the history of vaccine development, how it all began. Although inoculation, the practice of intentionally exposing someone to a matter from smallpox pustule in order to initiate a mild protective response to the disease against smallpox was practiced over 2000 years ago in China and India. A British physician, Edward Jenner, is generally credited the ushering into the modern concept of vaccination. In 1796, he used matter from cowpox vaccines to inoculate patients successfully against smallpox. In 1974, the WHO, that is World Health Organization, launched the Expanded Program on Immunization, EPI. In 1974, the goal was every child less than 1 year receive protection against 6 childhood diseases, that is tuberculosis, polio, diphtheria, pertussis, tetanus, measles. Tetanus toxoid vaccination protects the woman and their newborn. 1990, vaccination protected greater than 80% of the world children from the 6 main EPI diseases. New vaccines are continually being added to the EPI program in many countries. 1999, Global Alliance for Vaccination and Immunization extended reach of EPI, helped poorest country introduce new vaccine in national programs. Now, we will see what is vaccination. Vaccination is a simple, safe, effective way of protecting people against harmful diseases before they come in contact with them. Vaccine train your immune system to create antibodies just as it does when it exposes to a disease. However, because vaccine contains only killed or weakened form of a germ like virus or bacteria, they do not cause the disease or put you at risk of its complications. Most vaccines are given by an injection. Some can be given orally by mouth or sprayed into the nose. Now, how does a vaccine protect individuals and communities? When a vaccine person get vaccinated against a disease, their risk of infection is also reduced, so they are also far less likely to transmit the disease to others. As more people in a community get vaccinated, fewer people remain vulnerable, and there is a less possibility for passing the pathogen on from person to person. Lowering the possibility from a pathogen to circulate in the community protects those who cannot be vaccinated due to other serious health conditions from the disease targeted by the vaccine. This is called herd immunity. Who approves a vaccine? In countries where vaccines are manufactured, national or regional regulators oversee a vaccine development. This includes approving clinical trials, evaluating their results, and taking decision on licensing. In deciding, regulators must prefer to a very strict international standard on acceptable ethical clinical practice. Once a vaccine has been developed, national regulators decide whether to introduce a vaccine in their country. WHO provides information to support this process through comprehensive evaluation of the available evidence and its regularly updated position papers on vaccines. Now, this is a very important part. We'll just look at the types of vaccine. There are many types of vaccine categorized by antigen used in their preparation. Their formulation affects how they are used, how they are stored, and how are they administered. They can be categorized as mono or polyvalent vaccine. Vaccine may be monovalent or polyvalent. A monovalent vaccine contains single strands of a single antigen. Example is measles vaccine. Whereas a polyvalent vaccine contains two or more strands serotype of the same antigen. Example OPV. Combinational vaccines. Some of the antigens above can be combined into a single injection that can be that can prevent different diseases, or they can protect against multiple strands of infectious agent causing the same disease. Example, combinational vaccine against diphtheria, DPT, that contains diphtheria, pertussis, and tetanus antigen. Combination vaccine can be useful to overcome the logistic strain of multiple injections and accommodate for a child's fear of needles and pain. Now we'll look at components of vaccine. It has an active component, that is the antigen. It can be a virus, bacteria, or toxin. Adjuvants are added to enhance the immune response to a vaccine, like aluminum. Antibiotics prevents bacterial contamination, neomycin, gentamicin, polymyxin B. Preservatives prevents contamination. Example, repeated puncture of a multi-dose vaccine vial, like magnesium, thimerosal. Stabilizers protect the vaccine from adverse conditions such as the freeze drying process. It can be sugars like lactose, sucrose, amino acids like glycerine, protein like gelatin, albumin. trace components left over from vaccine production process like formaldehyde. Now we'll go to the important part, that is types of vaccine. It can be a live annotated vaccine, LAV, inactivated whole cell kill antigen vaccines, subunit vaccine, toxide vaccine, or the relatively new ones that are vector or DNA RNA vaccines. We'll discuss each of them one by one. Now, live annotated vaccine. Live annotated vaccine prepares from living microorganism, that is virus, bacteria, that have been weakened under laboratory condition. LAV vaccine will replicate in a vaccinated individual and produce an immune response, but usually cause mild or no disease, are derived from disease-causing pathogens. Like, for example, bacteria, it's tuberculosis, BCG, virus is oral polio vaccine, OPV, measles, rotavirus, or yellow fever. Inactivated whole cell kill antigen vaccine. Inactivated vaccines are made from microorganisms like virus, bacteria, or others that have been killed through physical or chemical processes. These killed organisms cannot cause disease. Immune response. Inactivated whole cell vaccine may not always induce an immune response, and the response may not be long lived. Several doses of inactivated whole cell vaccines may be required to evoke a sufficient immune response. For example, whole cell pertussis by bacteria and inactivated poliovirus, IPV. The third type is subunit vaccines. Subunit vaccines, like inactivated whole cell vaccines, do not contain live component of the pathogen. They differ from inactivated whole cell vaccines by containing only the antigenic part of the pathogen. This precise precision comes at a cost, as antigenic property of the various potential subunit of a pathogen must be examined in detail to determine whether a particular combination will produce an effective immune response within the correct pathway. They can be protein-based, polysaccharide, or conjugate. The fourth type is the toxide vaccines. A vaccine is made from a toxin, poison, that has been made harmless, but that elicits an immune response against the toxin, are based on toxin produced by certain bacteria, for example, tetanus or diphtheria. The toxin invades the bloodstream and is largely responsible for the symptoms of the disease, used as the antigen in the vaccine to elicit immunity. The fifth type is the viral vector vaccine. To carry the DNA in human cells. The DNA contained in the virus encodes antigen that, when expressed in the infected human cell, elicits an immune response. Recombinant vector vaccines work as a natural infection and are good at training the immune system to recognize and attack germs. They work by reproducing a living virus that has been engineered to carry extra genes from the germ infections infecting the body. The sixth one is the DNA or the RNA vaccine. In a cell, DNA is transcribed into RNA, which is then translated into a protein. A DNA or an RNA vaccine is essentially a subunit vaccine, but instead of introducing a subunit protein into the body, this vaccine provides specific genetic code, DNA or RNA, for the cell, which produce the subunit itself. RNA vaccine have the added benefit of no risk that the RNA will integrate into the patient's own genetic code, as could happen with the DNA vaccine. Now, how is the vaccine given? There are different routes of administration. We'll look into it. The routes of administration is the path by which vaccine or a drug is brought in contact with the body. It can be intramuscular, injection administer the vaccine into the muscle mass, as you can see, intramuscular in the muscle. Subcutaneous injection administered the vaccine in the subcutaneous layer above the muscle and below the skin. It's in the subcutaneous layer. And the intradermal is the dermis. And it also shows the angle at which these injections are given. Intradermal, administer the vaccine in the topmost layer of the skin. Oral administration of the vaccine makes immunization easier by eliminating the need for a needle and a syringe. Intranasal flu vaccine, intranasal spray application of a vaccine offers a needle-free approach through the nasal mucosa of the vaccine. This slide basically tells on the types of vaccine given by oral, intramuscular, subcutaneous, and intradermal route. Now we'll go into the vaccine trial. Vaccine can be prophylactic or therapeutic in nature. While prophylactic vaccines are given to normal participants, therapeutic or curative vaccine may be given to patients suffering from a particular disease. Many of the prophylactic vaccines are given to the pediatric group, that is the children. Now, how does a clinical trial take place? There are basically 4 types of phases. We'll discuss them one by one. If you go and look at the picture, it's clinical trial phase 1. What it does is test the safety immunogenicity of a vaccine candidate in a few low-risk individuals. Usually it is given in healthy adults to determine tolerability. Sample size is the number of people it is given. It's generally 10 to 100. And the detection of adverse effects, common or rare, it depends. And clinical trial phase 2 monitors safety, potential side effects, immune response, and determine optimum dose and schedule. It is given in 100 to 1,000 patients, participants basically. Clinical trial phase 3 address clinical efficacy in disease prevention and provide further safety information from more heterogeneous population and longer time of observation. It is given in 1,000 to 10,000 individuals. And then the submission is basically phase 4. The vaccine application is submitted to the regulatory authority, the government, for the approval to market. Introduction, when it's given to the public, involves making the vaccine available for use. We'll elaborate on each phases now. Phase 1 refers to the first introduction of the vaccine in a human population for determining of its safety, biological effects, including immunogenicity. This phase includes study of dose and routes of administration and should involve low-risk participants. The special features that you need to know: live annotated or kill vaccines pose concern about possible shedding of infectious agent, transmission to contact, and a possible reversion to a more virulent state. Therefore, volunteers, clinical trial participants are called volunteers because they're voluntary participating of such phase 1, trial require intensive investigation in a closely monitored clinical setting, including evaluation for any clinical sign or infection. Generally, phase 1 have their own phase 1 setup, and they are monitored in a closed environment, like like a hospital setup they have. Phase 2, it refers to the initial trial examining the effectiveness, immunogenicity, and dose range in a limited number of volunteers forming the target group, like children, adults, or those at risk of exposing to pathogen. Pharmacokinetics and the safety of the vaccine is also studied. Special feature is phase 2 trials can also provide preliminary information on protective efficacy through human challenges studies, whereas in healthy participants are deliberately infected with the pathogen. Such studies are commonly referred to as phase 2A studies and are appropriate only for a selective disease, whereas it is scientifically and ethically justified. Whereas the pathogen does not cause lethal infection and is not restrained to available treatment, and a complete and successful cure can be obtained. Phase 3, this focus on assessment of safety and efficacy in the prevention of disease involving controlled study on a larger number of volunteers in thousands through multi-centric different hospitals are there that conducts trials. These studies determine the protection offered by the vaccine and provide pivotal data for licensure. Efficacy in vaccine trials mean reduction in incidence of the disease after vaccination compared to the incidence that prevailed before vaccination. Effectiveness means information on protective rate conferred on a given population. Phase 4, that is the post licensure evaluation. These studies are done in the entire population or a subgroup to detect the rarer or unexpected events that may not be seen in smaller phase 2 or phase 3 studies. Post licensure studies of larger population in a more heterogeneous group of people over a longer period of time are necessary to provide ongoing assessment of vaccine safety and effectiveness. Key points that you need to know: vaccine are now, when in phase 4, are now in use for the general population and recipient are no longer monitored in a clinical trial. Spontaneous reporting is the cornerstone of most post licensure safety monitoring system. Post licensure surveillance, also known as post marketing surveillance, pharmacovigilance conducted after a product has been licensed and introduced for use in a population of vaccine safety is critical. Basically, you have to look into the safety as well as the efficacy even after the vaccine has been introduced in the population. Now we'll look at the COVID-19 vaccine candidates. We have already discussed there are different types of vaccines. We'll just look at the advantage and disadvantage in respect to the COVID-19. DNA and RNA. These vaccines use DNA or RNA molecule to teach the immune system to target key viral proteins. Advantage is easy and quick design. Disadvantage, never been done before. There are no licensed DNA or RNA vaccine currently in use. Existing example, none. Group testing these approaches, for example, Moderna and RNA, they are doing RNA, and Innovia, they are doing DNA. Ironically, these are the DNA and RNA vaccines. RNA vaccines are the first to be approved as of now. And live annotated vaccines, advantages are stimulates a robust immune response without causing serious disease. Disadvantage may not be safe for those with compromised immune system. Existing example: measles, mumps, rubella, chickenpox. Groups that are doing the COVID-19 uh, live annotated vaccines are Codagenics, Indian Immunologicals. Now, the inactivated advantage is safe because the virus is already dead and is easy to make. Disadvantages: it is not as effective. Some previous inactivated vaccines have made the disease worse. Safety for the novel coronavirus needs to be shown in clinical trial. Examples are like a polio vaccine, and groups that are testing COVID vaccines are Sinovac and Sinopharm. Now, subunit vaccine advantages: focus the immune response on the most important part of the virus for protection and cannot cause infection. Disadvantages: may not stimulate a strong response. Other chemicals may be needed to be added to boost long term immunity. Existing example: persuasive hepatitis B, human papillomavirus. Group testing these COVID drugs vaccines are Novavax and AdaptVac. Now, the viral vectors. Live virus tends to elicit stronger immune response than the dead response or subunit vaccine. Important to pick viral vectors that are truly safe. An immune response to the viral vector could make the vaccine less effective. Existing example: Ebola, veterinary medicines, and the group testing is University of Oxford and AstraZeneca, CanSino Biological, Johnson and Johnson. Now we'll just look at how long traditionally it took for a vaccine to be made. Traditional development, design and exploratory preclinical studies, and then process development preclinical toxicology studies. They are done in animals. It took 2 to 4 years. Then investigational new drug submission happened. Then the clinical trial in humans that happened. Phase 1 lasted for 1 to 2 years. Phase 2 for 2 years. Phase 3 for 2 to 3 years. Then biological license applications was a submission happen and for to the regulatory authority like FDA for the US or EMA for Europe. Then large scale production and distribution happened. So it took more or less 15 years or longer. But since the gravity of the situation of the COVID-19, it is happening on an expedited mode. You can see basically it's taking 10 months to 1.5 years total. Currently, as per the WHO draft landscape of COVID-19 candidates published on 2nd December, there are 51 candidates in clinical evaluation, that is clinical trial, and around 160 plus in preclinical. We'll look into each of it for to get a better understanding. Firstly, we'll look into Sinovac. The vaccine platform is inactivated. The number of doses is 2, and the timing of doses is 0 and 14 days. And routes of administration is IM, intramuscular, and the phases is phase 3. Wuhan Institute of Biological Products, Sinopharm, again inactivated, 2 doses, 0 to 21 days, intramuscular, phase 3. Beijing Institute, inactivated, 2 dose, 0 to 21 days, intramuscular, phase 3. Bharat Biotech, inactivated, 2 doses, 0 to 28 days, intramuscular, phase 3. University of Oxford AstraZeneca, non-replicating viral vectors, 2 doses, 0 to 28 days, intramuscular. CanSino Biological, Beijing Institute of Biotechnology, non-replicating again. It has 1 dose and it is intramuscular, again phase 3. Then there's Camellia Research Institute. It has 2 doses, non-replicating one, 0 and 21 days, intramuscular, phase 3 again. Gensine Pharmaceutical Companies. It has number of doses are 2. First dose is 0 and second is on 56 days. intramuscular, phase 3. Novavax, it is a protein subunit. Again, 2 doses, 0 to 21 days, intramuscular, phase 3. And Huizhou Institute of Microbiology, Chinese Academy of Sciences, protein subunit. 3 doses are there at interval of 0, 28, and 56 days, intramuscular again. Moderna, it's RNA, 2 doses at 0 and 28 days. It is intramuscular. BioNTech, Fosun Pharma, Pfizer one, RNA, 2 doses, 0 to 28 days, intramuscular. Medicago is 2 doses again, 0 to 21 days, intramuscular. Inovio Pharmaceuticals, DNA based, 2 doses, 0 to 28 days, intradermal. Beijing Wontal Biological Pharmacy, Xiamen University, 1 dose, intranasal. West China Hospital, protein subunit, 2 or 3 at 0 or 28 or 0, 14 or 28 days, intramuscular. Covert, RNA based, 2 doses, 0 and 28 days, intramuscular. Institute of Medical Biology, inactivated, 2 doses, 0 to 28, intramuscular. Phase 1, phase 1, phase 2. Research Institute for Biological Safety, Republic of Kazakhstan, inactivated, 2 doses, 0 and 21 days, intramuscular. Shenzhen, inactivated, 2 doses, intramuscular. Phase 1, phase 1, 2. Osaka University, it's DNA based, 2 doses, 0 and 14 days, intramuscular, phase 1, 2. Cadila Healthcare, DNA based, 3 doses, 0, 28, 56 days, intradermal. Genisen Consortium, DNA based, 2 doses, intramuscular. Kentucky, protein subunit, 2 doses, 0 and 21 days. Sanofi, protein subunit again, 2, 0, 21 days, intramuscular. Biological E, protein subunit, 2 doses, 0, 28, intramuscular. Israel Institute for Biological Research, replicating viral vector, 1 dose, intramuscular. Arcturus, it's RNA, and there is intramuscular doses is not mentioned. And Spy Biotech is VLP, that is viral like particle, 2 doses, 0 to 28 days, intramuscular. SimVivo DNA, number of doses is 1, oral. Providence Health and Services DNA, 2 doses, 0 to 28 days, intradermal. Codagenics is live annotated vaccine, number of doses 1 or 2, intranasal. Immunity Bio is non-replicating viral vector, 2 doses, 0 to 21 days, subcutaneous. And COVAX is protein subunit, 2 doses, 0 to 28 days, intramuscular. Chinese Academy, protein subunit, number of doses 2 or 3, and it is intramuscular. Merck, replicating viral vector, number of doses is 1, intramuscular. These are all phase 1. Institute Pasteur, replicating viral vector, 1 or 2 doses, 0 to 28 days, intramuscular. Imperial College London is RNA, number of doses 2, intramuscular. People Liberation Army Academy, RNA based, mRNA, 2 doses, 0 or 14 or 0 or 28, intramuscular. Generally, you've seen generally 2 doses are there. The reason being antibody concentration when first given, initial exposure to antigen, primary immune response is this. But when second exposure to antigen is there, The response is faster and response is larger. That comes, now we come to the end of the presentation. And for any diploma course or webinar related queries, you can contact our website or email ID. And we offer diploma in clinical research, pharmacovigilance, and clinical data management. You can like us on Facebook and subscribe on our YouTube, and we can have more productive webinars in the future. In case of any query that you might be having, you can always mention that in the comment section and we'll revert to that. Thank you so much for listening. Bye bye.